Very happy to be here. Thank you for the invitation. Very excited to be talking about my two favorite topics, therapeutics and diagnostics for snakebite envenoming. And today I will be focusing on the therapeutic part, specifically on current treatments, future therapeutics, and preclinical validation. So since this is an interdisciplinary summer school, I imagine that you guys have a lot of different backgrounds, so I'm going to start a bit basic and then get into some of the specifics. But if there's anything that's not clear, please just ask me questions after I'm done with the presentation. Right, so a bit simplified, but very broadly speaking, you can split therapeutics for snakebite into two types, the specific and the unspecific or supportive. So what I mean with this is supportive treatments, that could be something like antibiotics to combat wound infections or artificial respiration. If, well, you probably know that some snake venoms have neurotoxins that can paralyze the respiratory muscles, leading victims to be at a danger of asphyxiation. Well, then you might need something like artificial ventilation to as a kind of treatment for that. But there are, of course, also specific treatments. And with this, I am thinking of antivenom, and that is what we're going to be spending most of the time on today. So in case some of you are kind of new to the field, I'm just going to briefly run over how antivenoms are produced. So very briefly, you start by milking a venomous snake for its venom, or potentially several different venomous snakes, if you want to create a polyvalent antivenom that can neutralize many different snake venoms instead of a monovalent antivenom for just a few snake venoms. You then take this venom and use it to immunize an animal. Often people immunize horses. This is mostly a practical matter. Horses are, they're more docile than tigers. They're bigger than rabbits, and they don't have the same sort of religious problems as, say, cows or pigs might. But it really could be whatever animal. Point being that once you immunize this animal with the snake venom, the animal's immune system will recognize the immunogenic venom components as foreign and start generating antibodies towards them. These antibodies will then be found in the animal's blood. So you can extract blood for the animal, and just like when humans donate blood is not something that kills them, you just take a portion of blood, and from that you can purify these antibodies. You can then further affinity purify them or treat them with enzymes if you want the antibodies to have different formats. And then you can formulate them, pour them in a bottle, and that's essentially what an antivenom is. Just antibodies from a hyperimmunized animal. So, is this perfect? Well, of course not. I'll go as far as to say there probably isn't any one perfect therapeutic. But antivenoms are amazing in that they save a lot of people's lives, and we would be terribly off if we didn't have them. That being said, there are, of course, still different areas of antivenoms that could be improved. So I'm going to run through some of the most common points of criticism for conventional antivenoms, but please do bear in mind that all antivenoms are not the same, and antivenoms come in a lot of different qualities. So one problem that some antivenoms suffer from is that only a low percentage of the antibodies in that antivenom actually recognize venom toxins. This is, of course, this is because when we immunize animals to create antivenoms, we use immunologically mature animals whose immune systems have already been exposed to a plethora of other pathogens and therefore have already produced antibodies against all those other different pathogens. So if you, as some antivenom manufacturers do, just take all the antibodies that you can from the blood of that animal, well, then only a small percentage of them will actually be directed towards venom toxins. The rest will be directed towards whatever pathogen the animal has been exposed to. You could get around this just by affinity purifying antibodies against venom toxins, but not everybody does this. Another common point of criticism is that some antivenoms are prone to induce adverse reactions. This can be due to suboptimal quality of manufacturing, but there is also an inherent risk to using animal proteins because, I mean, once you administer those animal proteins, those animal antibodies to a human being, there's always the risk that our immune system will recognize these proteins as foreign and mount an immune response against them. This also has the unfortunate side effect that you can actually become allergic to antivenom, which could be problematic if you need it more than once in your lifetime. Also, clearly, animals are not machines, so you can't have two animals that will produce the exact same antibodies for an antivenom, and this might also lead to some batch-to-batch variation. It can also be expensive to keep horses and to keep snakes for the antivenom production, and this can in turn make the antivenom expensive and even unaffordable. And this is very problematic because statistically there is a correlation between snakebite envenoming and poverty. So really, for any solution you want to develop for snakebite envenoming, you need to keep the end user in mind. You need to keep in mind that this has to be affordable. Otherwise, technologically, it might be a marvel of the world, but it will just never, ever help anyone. Also, immunizing an animal is somewhat of a time-consuming procedure. I mean, very similar to when we vaccinate humans. I think probably the last thing I was vaccinated against was hepatitis for traveling. That was also something like, you get an injection, you wait 6 weeks, you get another injection, you wait half a year, you get another injection. It just takes time for the immune system to produce good antibodies. And that means it takes time to produce a good antivenom. And this can be problematic because it means you can't just quickly produce more antivenom, even if you urgently need it. And all of these factors combined just can mean that antivenom is sometimes unavailable where it's needed the most. And this is, of course, highly problematic. But scientists, being a very optimistic bunch of people in general, really wanting to improve the world and explore their curiosity, are, of course, working on different ways to improve antivenoms. And there are different sort of veins of research when it comes to this topic. One is simply to improve current antivenoms. That could be by improving the immunization mixtures and methods. For instance, maybe we can be smarter with the specific venoms or toxins we immunize these animals with. Maybe there's something about the immunization schedule, like the frequency of immunizations. Or maybe we could purify the antibodies better afterwards or have an even higher standard for manufacturing. All of those things would likely help make the antivenoms better. Another vein of research is just, scrap that, let's make something completely new. And I think the two main things that are getting attention in that field, as far as my opinion goes at least, is recombinant antivenoms and small molecule therapeutics. Now, just briefly, recombinant antivenoms are antivenoms comprised of recombinantly expressed antibodies. So, in short, antibodies that have been designed and expressed in the lab. Small molecule therapeutics are, as the name indicates, small molecule therapeutics. They're often drugs that have previously been tested for different indications, and they often target enzymatic toxins, which is actually really interesting because that can mean that they can neutralize a bunch of different toxins at once, as long as those enzymatic toxins all have similar active sites that the small molecule therapeutic can then interact with. And finally, there is the idea that, okay, how about we just combine all of the above? For instance, there are some toxins that are just difficult to raise good antibodies against using conventional immunization methods, simply because even though those toxins are very medically relevant, they're just very poorly immunogenic. So one idea is to use recombinant antibodies against those poorly immunogenic toxins and spike those recombinant antibodies into traditional antivenoms to sort of catch out the holes, like the toxins that are not well covered by the traditional antivenom. Another idea is that a lot of these small molecule therapeutics could be made orally available. So potentially you could administer those small molecule therapeutics in a pre-hospital setting, and that could maybe then buy snakebite victims time to actually get to the hospital and get a traditional antivenom. So there are a lot of potential synergies here. But of course, it won't fully be possible to explore them until any of these new types of treatments eventually hit the market. So again, just in case some of you are new to the field, I would just want to quickly sort of summarize the general pros and cons of these different approaches. This is just my take on this. And of course, different researchers might prioritize different pros and different cons. But still, I thought maybe it could be helpful. So for improved traditional antivenoms, in my opinion, the greatest advantage of them is that I think they're very likely to succeed. It's essentially just doing what we're already doing, but doing it slightly better. And I don't see why that wouldn't work. And I also think this is something that could potentially benefit snakebite victims quite soon. It's more of a short-term thing to make these small improvements and tweaks. I think the main disadvantage is because it's mainly doing what we already do, but better, we are only likely to see incremental improvements from this type of research. For recombinant antivenom, I think one super interesting aspect is that you can just, instead of making animal-based antibodies, you could just make human antibodies, which will be compatible with the human's immune system, and therefore potentially be safer to administer. They'll be less immunogenic. Also, it is way faster to produce antibodies in a big cell tank in a lab than to go through a whole immunization procedure. So you could potentially produce these recombinant antivenoms a lot faster once they have been developed. Also, I think it's very nice that there's this opportunity to have a defined mixture of antibodies where you know that every single antibody in an antivenom actually targets a highly relevant toxin in the venom. But it's not like recombinant antivenoms are the perfect solution either. First of all, it takes a really, really long time to develop any new antivenom products, which also means that probably no snakebite victims will get any benefit from this for the next several years. This is like a long-term project. Also, while I think this all sounds reasonable and realistic, because all of these different recombinant antivenoms are still being developed, we still don't have any like clinic evidence that they will work. We have preclinical stuff in mice, but that isn't exactly the same thing. And finally, the small molecule therapeutics. One thing that's a big advantage is that a lot of these are repurposed drugs that have previously been tested for a different indication. And that also means that a lot of them have previously gone through phase 1 clinical trials. So we already have safety data on them. Plus, this means that they probably won't have to go through phase 1 clinical trials again. So they potentially have a shorter time to market. They could help people sooner. A lot of them also have the potential for being delivered orally, like a pill or something, which could help bring treatment closer to where the bites happen and really help buy people time, because with antivenom, generally, the sooner you get it, the greater your chances of making it out alive, and with very few comorbidities are. But yeah, so maybe you could just like help treat people sooner and buy them time till they get the proper antivenom. Also, a lot of these small molecule therapeutics are likely to be quite cheap to produce. The main disadvantage of them, at least in my opinion, is that while each of them can sort of fairly broadly target a toxin type or subtype, they only do target that one. So I don't think there's any one small molecule therapeutic that's likely to just cure snakebite entirely. I think they will likely always need to be used either in combination with other small molecule therapeutics or with some other kind of treatment. Anyway, this is just the overview. And I think on Friday, Juliette Bory will give you like the whole full overview of everything that's going on in the research pipeline right now. And I imagine Charlotte Dawson is going to be talking a lot about small molecule therapeutics. At least they do a lot of amazing work on those at the Liverpool School of Tropical Medicine, which is perfect for me because that leaves me to talk about recombinant antivenom, and that's what I have worked a lot on, especially in the past. So, as I said, recombinant antivenoms are based on recombinant antibodies. So really, the whole trick to getting the best possible recombinant antivenom you can is to have the best antibodies you can. And there are different things you need to consider in terms of having fit-for-purpose antibodies. One thing is the antibody format you're going to choose. So here on the left, I've just listed a bunch of like fairly commonly used antibody formats, such as IgGs and F2s. And on the right, I've listed some of the crazier formats that people have looked into, just to say that there are other options than the usual suspects when it comes to choosing the antibody type you want to work with. And when you choose the type or the types, there is a lot of things to keep in mind, such as the pharmacodynamics of the specific antibody format, like how well can this neutralize a toxin, the pharmacokinetics, so what's the distribution of this antibody in the body going to be like, how long does it persist in the body before it's degraded or excreted somehow. You also need to consider the origin of the antibody and what this origin means in terms of the antibody's safety and propensity for inducing adverse reactions. Different antibodies can also be produced in different ways. Generally, smaller antibodies are easier to produce, which could also make them cheaper to produce. And again, you need to really keep affordability in mind for these things. And you also need to consider how the pharmacokinetics of the specific antibody you want to use fits the toxicokinetics of the specific toxin you might want to target. So choosing the right format or formats is really important. Another thing that will likely be very important for recombinant antivenoms to ever make it into clinical use is obtaining broad neutralization. So what do I mean by this? Well, every single snake venom contains a lot of different toxins. And if you needed a single antibody to neutralize each of these toxins, that would end up being a lot of antibodies. That's what I've tried to show on the left side of the screen here. So if you need that many antibodies, that's going to be very technically challenging, and it's likely going to be very expensive. So that's not a good solution. So what people are looking into instead is creating broadly neutralizing antibodies. So antibodies that can neutralize multiple different toxins from multiple different snake species. But how do you get broadly neutralizing antibodies in order to make your recombinant antivenom more technologically and economically feasible? I'm gonna give you 3 different examples of how this could be done. The first one is with the creative cross-panning strategies. So essentially, a lot of recombinant antibodies are discovered via display technologies such as phage display. That's what I have a schematic of here. And there are a lot of different cool ways you can do phage display to increase the likelihood of achieving broadly neutralizing antibodies. If you want to know more about this, please ask me in the question session, because I don't have time to explain the whole thing here. What I do want to do instead, though, is show you an example where some of my colleagues did just this. So I have a colleague who tried having these creative approaches to phage display and using them to raise a broadly neutralizing antibodies against long-chain alpha neurotoxins. And what you see here on this slide is her testing the antibody she found against different whole venoms in pre-incubation assays. So essentially, you will see survival of the mice that were injected, and then the time that they survived. So on the left here, I don't know if you can see my mouse. I hope you can. But essentially, we can see some mice that were injected with just pure Goethe-Universität venom, or pure venom plus a random IgG, and we can see that those mice died very fast. However, mice that were instead injected with either venom plus antivenom or venom plus the antibody she found, they actually survived for the full 50-hour duration of this experiment. So this is an example of a single broadly neutralizing antibody that actually neutralized a whole venom from a snake, which I think is kind of crazy and very promising. However, when this single antibody can neutralize the whole venom of Goethe-Universität, that is also because Goethe-Universität has a venom that is comprised largely of long-chain alpha neurotoxin. So here's another example, Ophiophagous hana venom, which is a bit more complex in its composition, let's say. And here, when she injected the mice with the venom plus her antibody, she saw, well, not full survival, but at least a prolongation, like prolonged survival. So I think this is a very nice indication that you can actually achieve these broadly neutralizing antibodies. She also tested it in rescue assays and a bunch more snakes, but you will have to read the paper if you want to see those results. Okay, so cross-panning is one strategy for obtaining broadly neutralizing antibodies. A very different strategy is using consensus toxins. So consensus toxins are sort of artificial toxins. It's essentially where you take the sequences of a bunch of different toxins of your choice, and then you find sort of the average sequence based on those, and you express that average sequence, which then is the consensus toxin. So some of my colleagues did this. They designed and expressed consensus toxins, and then they raised antibodies against those consensus toxins. Again, I don't have time to show you all the data on how these consensus toxin antibodies actually bind to a bunch of different natural toxins. I will just go straight to a neutralization assay. So what you can see here is that they're trying to target an enzymatic toxin. So out here on the y-axis, you can see the activity on the enzymatic toxin, and on the x-axis, you can see the concentration of antibody they added. So if they add a totally random antibody shown in some kind of turquoise green color here, that antibody doesn't affect the activity of the enzymatic toxin at all. But when they added either of two different antibodies raised against a consensus toxin, they could actually see that those antibodies inhibited the activity of the natural toxin, which is also very cool. So potentially you can use these consensus toxins to generate antibodies that neutralize hopefully each of the individual natural toxins that were part of the design of the consensus toxin. A final thing you can do to find broadly neutralizing antibodies is use machine learning. So this is a very, as far as I'm aware, new area of research, but I think it's very promising. So what some people are trying to do is essentially generate a bunch of data on, well, generate a bunch of antibodies, generate a bunch of data on how well those antibodies bind different toxins, so essentially a bunch of data on antibody affinity, then get the sequences of all those antibodies, then at the same time create structural models of how those antibodies might interact with the toxins, and then feed all of this data into a machine learning algorithm. What they then hope to achieve is to have this machine learning algorithm be able to sort of make a link between a given antibody sequence and the likely affinity of that antibody for a specific toxin. So that in the future, you can do the whole thing in reverse and say, okay, I have toxin 1 and toxin 2. Now please generate an antibody sequence that will bind to both those toxins. Which, if it ends up working, I think could be incredibly useful, because we could skip a whole bunch of antibody discovery and essentially just tell this algorithm, well, give me something that works for X, Y, and Z. So that would be very cool if it works, but it's still early days with this research. Okay, so we've talked about improving antibodies, both by making sure to choose the right format, different ways of achieving broad neutralization. A final thing I just want to touch on is pH sensitivity. And this is going to be a bit brief. But essentially, there is a theory that some antibodies might actually bind to toxins so well that they prolong the toxin's presence in the body. They just make sure that the toxin stays with the antibody and gets cycled around the bloodstream. So one way to potentially avoid this issue is to engineer pH-sensitive antibodies. So antibodies that will bind to toxins in the neutral pH of the bloodstream, but then let go of the toxin they bound in the more acidic environment in endosomes. In theory, this should mean that the toxins are then sent for lysosomal degradation, while the antibody is recycled via the FcRn receptor, and the whole thing can go another cycle. So this is just one idea some people are looking into for how to create even better antibodies as well. Okay, so let's say now you have your new snakebite therapeutic, whether it's more of a conventional antivenom, recombinant antivenom, small molecule therapeutic. No matter what, you will have to test it preclinically and figure out if it actually works before you ever get to clinical trials, and therefore before you can ever help anyone. So how can you do this? Well, one very commonly used method is the pre-incubation assay. So essentially an assay where you mix the venom with your treatment and then inject mice with that and monitor survival. This is, of course, not a very lifelike scenario, but it is useful because it allows the treatment to neutralize the venom before it's injected into the mouse. And if the treatment doesn't work even under these conditions, it's not very likely to work at all, and you can save yourself a lot of trouble, and you can save a lot of mice a terrible death by just kind of calling it then and there. If, however, your pre-incubation assay results look promising, what people often do is go to a rescue assay, a more lifelike assay where first you inject the mouse with a venom, simulating a snakebite, and then after a pause, you inject it with your treatment, simulating receiving, well, treatment antivenom. And again, you monitor survival. And it is actually really important to do both these assays, and I'm gonna give you an example why. So my colleague, Chris, was trying to discover one of these neutralizing antibodies. And he had a good sense from in vitro assays that it was working very well, so he went to check it in mice. Very quickly about this graph, let's just say like essentially the y-axis, the higher the bar, the worse the mice are feeling. So what he saw in his pre-incubation assay was, when he added just venom, this kind of striped bar, the mice were not doing well. But when he added venom plus his antibody called B12, the mice were doing a lot better. Great, thought Chris. Very promising pre-incubation assay. Now to do a rescue assay and see just how good my antibody actually is. But then in the rescue assay, again, giving just venom, strike bar, mice were feeling terrible, but actually now giving venom plus the antibody, the mice felt even worse. So in the pre-incubation assay, it looked like his antibody was neutralizing the snake venom, but in the rescue assay, it actually looked like his antibody was potentiating, like enhancing the snake venom, which is why you really have to make sure to do both assays because you can get wildly different results. He just got this paper describing this finding accepted for publication in Nature Communications yesterday. So I would urge you all to go read it, but if you have follow-up questions, we can also discuss it during the break. All right. Of course, though, in general, we are trying to move more away from doing animal experiments and to test the therapeutics, all the different ways we can think of in vitro before ever getting close to mice, in line with the 3 R's of animal experimentation, so replacement, reduction, and refinement. So here at the end, I just wanted to briefly mention some of the other methods that we're using. So one is a patch clamp, which we find especially useful for neurotoxins. Essentially, you can measure the current across a membrane in the presence of either a neurotoxin, which, you know, a lot of them work by opening the ion channels and letting all the ions go through, so you have a current. So either you test just in the presence of a neurotoxin or in the presence of the neurotoxin and your therapeutic. And then you see if the therapeutic has altered the neurotoxin's activity. Very similar, we do a lot of cell viability assays just to see, okay, if we co-administer a treatment with the venom, can we keep the cells from dying? And of course, there are a lot of enzymatic assays you can do, which might be especially useful for hemotoxins. Okay, I talked a lot now, and that's really all I had to say for now. I will return terribly with diagnostics on Friday, but I would love to take some questions on this right now.